All right, I think 8:01, we'll go ahead and get started. So welcome everyone, and thank you for joining us for the John Nicholls Endowed Lecture at the Marine Biological Laboratory. Our speaker today is Vanessa Ruta, and will be joined by Stephanie White and Alberto Pereda, the course directors of the Neural Systems and Behavior course. While we remain restricted to having a limited in-person audience, we're excited for this opportunity to bring pieces of the MBL Advanced Research Training courses to the public through the virtual Endowed lecture series. This series is comprised of 8 lectures throughout the summer from 8 different courses, and we encourage you all to register for as many as you're able to. Over the years, generous friends and family have honored eminent scientists and loved ones with 26 endowed lectures at the MBL, and this series would not be possible without that support. If you're interested in learning more about this opportunity, please email development@mbl.edu. We have a couple housekeeping items to go over before we get started. So to remind everyone, this is a Zoom webinar, and we will have a dedicated question and answer section at the end. So if you have any questions, please submit them in the chat in the Q&A function below, and we'll get to them at the end. So without further ado, please welcome Stephanie White and Alberto Pereda.
Hello, welcome to the 2021 John Nicholls Lecture, which this year is broadcasted virtually. Alberto?
Welcome. This John Nicholls lecture has an special event for many years in SMB, but this year it's going to be followed by the first town hall ever, Neurosystem and Behavior, which is going to be virtual, attended, I hope, for many of you, which are part of the NSMB community. So the town hall is going to follow the lecture, so stick with us after the lecture, because we're going to see something very fun. So I think we going to see a few words about John Nicholls. John Nicholls received his MD from Charing Cross Hospital and a PhD from the Department of Biophysics at University of College London. In addition to University of College London, he worked at the universities of Oxford, Harvard, Yale, Stanford, and Basel. Since 1998, he has been a professor of neurobiology at the International School for Advanced Studies in Trieste, Italy. During his PhD training in Bernard Katz's lab, he investigated the properties of denervated skeletal muscle and sensory transduction. Together with Stephen Koffler, he later studied interactions between glia and neurons and the functional effects that follow changes in the extracellular concentration of potassium. But perhaps John Nicholls is especially known for his studies on regeneration in the nervous system after injury. This work focused on the properties and functions of identified neurons in the lead, particularly on the way in which they regrow to restore function after injury. His commitment to education is an integral part of his scientific persona. He's well known for his textbook From Neuron to Brain, which is already in his 5th edition. He's also known for his involvement in many teaching institutions, such as the MBL, and for leading many outreach programs, including schools of neuroscience in Latin America. Finally, I would like to say that none of this achievement would have been possible without his genuine, warm, and inclusive personality. So without further delay, I now invite 2021 NSMB student Molly Maddy to introduce the speaker of this year's John Nicholls Lecture, Dr. Vanessa Ruta.
Hello, friends, and good evening. I'm Molly Maddy, a postdoctoral research fellow at the Salk Institute and current NSMB student. I'm delighted to have the opportunity to introduce our speaker for this evening's John Nicholls Endowed Lecture, Dr. Vanessa Ruta. Dr. Vanessa Ruta is the Gabriel H. Ream and Herbert J. Caden Associate Professor in the Laboratory of Neurophysiology and Behavior at the Rockefeller University in New York City. A true New Yorker, Dr. Ruta received her BA in chemistry from Hunter College and then did her PhD and her postdoc at the Rockefeller, where she now runs her independent lab. From her CV, the only evidence that Dr. Ruta has ever left New York City were her 2 stints at the Hole. As an undergraduate, Dr. Ruta attended summer research programs here, and she also attended the neurobiology course here as a graduate student. Dr. Ruda has earned numerous fellowships and awards, including the NSF-GRFP as a graduate student, the Helen Hay Whitney as a postdoctoral research fellow. She's a Pew Scholar, McKnight Scholar, NIH's New Innovator Award, Simons Foundation Collaboration for the Global Brain Investigator, and was recently named a MacArthur Foundation Fellow, among many other accolades. She also maintains an excellent record of mentorship and support that is evident not only in a recent Distinguished Teaching Award, but also in the confidence and passion that we have observed in her trainees, two of whom we've actually gotten to know over these past few weeks. We had the pleasure of meeting Dr. Ruda over lunch today, and we all enjoyed her calm presence, engaging questions, and honest discourse about doing research that doesn't always come easily and might be uncomfortably difficult at times. A trained dancer, Dr. Ruda explained that science actually came more easily to her than dance. So it was dance that taught her to stick with hard problems that have little immediate and visible rewards. Dr. Ruda's lab is currently investigating how stimuli in the physical world shape the function of neural circuits and how those are translated into innate and learned behaviors. She uses multidisciplinary approaches that encompass many of the skills that we have been learning here at NSNB: electrophysiological recordings, functional calcium imaging, and quantitative behavioral assays. Today, she will talk to us about one of the many projects ongoing in her lab about the basis of courtship in Drosophila. Please join me in welcoming Dr. Vanessa Ruda virtually to the NSNB John Nicholls Endowed Lecture here in Woods Hole.
Thank you so much, Molly. That was a really lovely introduction and very generous. Let me just share my screen and then I'll start. Okay, so I first of all want to just say how pleased I am to be here today. I want to start off actually by congratulating you and your classmates and all the faculty of NSNB for restoring it to an in-person format, which I'm sure was actually no small feat, but actually completely worthwhile from what I heard from the students today. I wish I could be there with you, but I'm actually very honored to be here to give the Nicholls lecture virtually, and certainly to participate in this sort of virtual symposium for NSNB. And as you heard, actually, I did not take NSNB, but actually I took the neurobiology course from the year 2000, along with one of your illustrious co-directors. And I should say that this was a completely transformative experience, as all my time in Woods Hole was. And over the past 21 years, it's hard to believe it's been that long, I would say that my really work has become increasingly focused in the realm of trying to understand the neural underpinnings of behavior. So I'm particularly pleased to be a part of this lecture series. And I am also very happy that some of my students, including one of my wonderful students, Chad Morton, is actually in the course this year and continuing the tradition. So let me just get my pointer and then I'll begin. I want to tell you today about some of the ongoing work in the lab, where we've been thinking about how animals really display an extraordinary diversity of behaviors, both within and between different species. And my lab has actually been really interested in thinking about how neural circuits can adapt over these very different time scales. So one long-term focus of the lab has been to understand how neural circuits can adapt rapidly through learning to allow every individual within a species to appropriately respond to their unique experience of the world. And I think just as a field in neuroscience, we've begun to gain some real kind of mechanistic insight into how these rapid forms of adaptation can give rise to behavioral variation amongst members of a species. But we know far less about how neural circuits are adapted over these much longer time frames, over evolutionary epochs, to give rise to the incredible diversity of behaviors that exists across different species within the animal kingdom. And I should say that in part, this is due to the fact that studies of behavioral evolution have often focused on the genetic mechanisms that could explain behavioral variation. But of course, genes do not specify behavior, but rather encode a blueprint for the assembly and function of neural circuits through which behavior is expressed. So neural circuits are therefore really poised to serve as a central substrate for evolutionary selection. However, until now, it hasn't been possible to examine brain circuits at a resolution that could reveal how they've been shaped by evolution to generate novel behavioral traits. So about 6 or 7 years ago, my lab began to develop methods to compare the same behavioral circuits across closely related Drosophila species, allowing us to begin to address this question by pinpointing the evolutionary adaptations that give rise to species-specific behaviors. And so the circuits that we chose to focus on are those which underlie male courtship behavior in Drosophila. So in flies, males court females with this really lengthy and elaborate ritual in which a male orients towards a female, taps at her abdomen with his foreleg to sample pheromone cues that cover her body. And if this sort of discrete gustatory assessment indicates that she's an appropriate mate, as it obviously has in this case, he will persistently pursue and sing to the female by vibrating a single wing to generate a species-specific courtship song. So one of the really remarkable aspects of this behavior is the fact that it is really rapidly evolved across the nearly 2,000 members of the Drosophila genus to become fantastically diverse, even between very closely related species. And I think a beautiful and kind of representation of that is the fact that Drosophila suzukii is actually a close cousin to Drosophila melanogaster, where males have wings that are adorned with pigmented spots. And so they're rather than follow the female from behind and sing to her, Suzukii males orient towards the front of the female and perform a silent visual display. So how did evolution tweak with the same ancestral circuits to generate such striking behavioral variation? Of course, divergent behaviors between species could arise from differences in sensory input, variation in the anatomic or functional properties of conserved neural circuits, or the evolution of completely novel behavioral pathways. And we can begin to differentiate between these and other mechanisms really by leveraging the enormous behavioral diversity exhibited across this genus, where actually, of course, the courtship rituals of Drosophila suzukii and melanogaster really represent just two snapshots of multiple independent evolutionary outcomes. And so if we look across this genus, males of closely related species, these courtship behaviors have diverged at multiple levels. At the level of sensory input, males of each species use a distinct complement of excitatory and inhibitory cues to identify an appropriate mate. And at the level of motor output, each male has really evolved a kind of distinct repertoire of motor displays that they perform to persuade their conspecific females to copulate. And I should say that the rapid evolution of these behaviors has really been likely driven by the fact that many of these species co-inhabit within the same environments, where they often meet each other and mate on the same patches of rotting fruit like this mango. And yet they remain reproductively isolated. That is to say that copulation rarely occurs, and when it does, it produces sterile or inviable offspring. So clearly there has been strong selective pressures to generate morphological and behavioral signatures of species identity to prevent this futile copulation. Okay, so despite the extensive diversification of courtship behaviors, what I'm going to tell you today is that there really is a common node within the courtship circuits across members of this genus, which reflects the fact that fundamentally males of all species have to perform the same basic computation that is essential for their reproductive success. That is to be able to identify an appropriate female mate and become highly aroused and motivated to pursue her until consummation. And so what I'll show you today is that we've been, first I'll tell you about how we've been taking advantage of the really rich genetic tools that are available in Drosophila melanogaster to begin to understand how a male's sexual arousal controls his faithful visual pursuit of a female during courtship. And this is the work that was spearheaded by a graduate student in the lab, Tom Heinmarsh-Sten, with the help of another graduate student, Rufei Li. I'll then describe how we've been actually exploring how these same kind of courtship circuits have been tailored to species-specific needs, both the sensory level of sensory preferences and motor displays. And this is the work of two postdocs in the lab, Rory Coleman and Adrian Otopelek, who of course was actually part of the course teaching in the course this year, and it has been really helped along by senior research scientist Nessa Morante. And I should say that by taking this sort of comparative approach, we've gone to start to gain some early insight into kind of the mechanisms of behavioral evolution. Okay, so over the past few decades, I would say Drosophila courtship has really emerged as a powerful model for studying the expression and regulation of internal arousal states, because it really represents a striking switch in male behavior. So if you introduce a male to a female, he's really indifferent to the presence of the female, even despite occasional encounters, as you can see here, until a discrete moment in time when he identifies her as an appropriate mate and becomes aroused, after which he will faithfully track and sing to her, actually for many minutes, until either copulation occurs or in nature she would decamp. And I think that if you just simply plot the distance between male and females over time, you can appreciate that the onset of male courtship really represents a kind of temporally sharp transition into an enduring behavioral state, where males go from being initially apathetic to the presence of the female and maintains large distances between her and him, to actually following her intensely and pursuing her with great intensity as an object of desire. So work from a variety of different labs over the past few years has actually identified a population of only 20 neurons which are contained within the male brain. These are male-specific neurons called P1 neurons that we believe mediate this switch into this enduring arousal state. These neurons are thought to integrate from multimodal sensory signal or pathways to really represent the sexual suitability of a potential mate, and their activation can trigger enduring courtship displays. And so in past work, we actually previously traced excitatory and inhibitory pheromone pathways to their point of convergence at the level of the P1 neurons to understand how the combined input of these pathways really tunes mate preferences. And so, for example, we can image the P1 neurons in male flies that are walking on an air-supported ball, like a little tiny fly-sized treadmill, and then bring in the abdomen of a female, allowing him to basically sample her olfactory and gustatory bouquet. So here you can see as we image the P1 neurons, the activity is low until the male basically taps and tastes the pheromones on his conspecific female. And you can see that the activity of P1 neurons are strongly activated every time the male fly, the melanogaster male, actually tastes the pheromones on a melanogaster female. But if you taste the pheromones on females of other species that carry different pheromones, you can see that the activity is either much lower or even there's some level of potential inhibition. And so this really highlights how the P1 neurons are actually tuned to pheromones of an appropriate mate. I also mentioned that these neurons we know are able to trigger in persistent courtship displays. And this has actually been beautifully demonstrated by a variety of different methods. But the approach that we took was to take advantage of the really exquisite genetic tools in Drosophila to express the light activated ion channel CS crimson just in the P1 neuron population and provide males with a slowly rotating magnet. And you can see that initially the male is basically, again, really indifferent to the presence of the magnet as it rotates around the arena. But then after even a very brief optogenetic illumination of this population, the magnet becomes intensely attractive to the male, and he will actually persistently pursue the magnet and sing to it, despite the fact that there's obviously no appropriate behavioral feedback. And so this raises this really interesting question: how is it that the salience of visual cues is really transformed by a male's internal arousal state, controlled by the P1 neurons, to give rise to this sort of persistent courtship pursuit? And so to address this question, we took advantage of a system that was developed in the laboratory, a kind of closed loop tethered system, which is where we have basically a visual panorama with a dot projected onto the screen, which goes back and forth. And actually, I realize I'm missing the little schematic of this panorama. But what I'm going to show you again, as I go back to the slide, is the fact that actually you can see now here is just the shadow of a dot that goes back and forth. And one of the real advantages of this system is the fact that we can completely uncouple this visual dot that moves back and forth from the male's internal arousal state. So basically his sensory signals and his internal arousal state are completely uncoupled in this system. And so what you just saw is that the male basically, as soon as the P1 neurons are activated, the male begins to track this visual target. And he does so with really remarkable fidelity, turning left and right with every sweep of the visual dot as it moves back and forth within his visual field of view. All the while, he performs the ipsilateral wing extensions that really are a hallmark of the production of the courtship song. And so we can quantify this behavior by actually defining a tracking index, which reflects both the fidelity and the vigor of a male's turning behavior over time. And so if we look at how actually the kind of this kind of tracking and chasing behavior unfolds, we can actually, what we can do is basically transform this into a raster kind of plot over time, and then align many cycles. And what you can see is that even 3 seconds of activation of the P1 neurons to this visual dot that goes back and forth drives this long-lasting pursuit over many, many minutes, providing really a beautiful overt behavioral readout of this switch into an enduring arousal state. And indeed, some males were actually willing to track this visual dot for up to 24 meters, which is as if a human chases partner for 16 kilometers, really underscoring how this is a highly motivated state of pursuit. Okay, so persistent courtship can be triggered by exogenous activation of P1 neurons, but it could also be induced by allowing males basically to sample the pheromones of a conspecific female and mimic his initial sensory assessment of a prospective mate. In addition, by modifying the visual stimulus to more closely replicate the natural statistics of female motion in free courtship, we found that this visual stimulus alone was actually sufficient to evoke persistent courtship pursuit. And one of the things that this really kind of striking observation reveals is that in the absence of any other additional cues, males perceive a moving target, a dot that goes back and forth, as a female and are really primed to court this fictive female over time. So independently of how males initiated courtship, one interesting feature of this behavior is it doesn't appear to represent a binary switch. That is to say that even though transient activation of the P1 neurons drives this really persistent change in behavior where males will track the visual target for many tens of minutes, on a much more rapid time scale, if you look at the tracking index, actually their vigor and fidelity of a male's courtship pursuit fluctuated over time and was even punctuated by brief pauses during which he stopped tracking the visual stimulus despite its continued presence, before then rapidly reinitiating. And what this indicates to us is that courtship is really regulated over multiple time scales. On the one hand, there's this long-lasting change in male behavior that lasts for many tens of minutes to underlie the sort of faithful pursuit of a female. But it's also regulated on a moment-by-moment time scale to allow for putatively to allow for flexibility in his behavioral response. And so we believe we have elucidated really a concise model of a visual motor circuit that can explain both the robustness and flexibility of courtship. First, it starts to highlight how P1 neurons, once they're activated, drive males into a state of sexual arousal, which gates the way visual signals are propagated to the motor systems to underlie the high fidelity pursuit of a female during courtship. But a second feature of this circuit, which is really critical, is that it is under constant regulation from the P1 neurons to assure that actually males continue to track appropriate targets, even in the context of more complex social scenes like the one I showed you on a rotting mango, where there are many equivalent visual targets. And so I'm just going to highlight a few of these observations that support this basic model. Okay, so in thinking about how visual signals can be transformed by a male's arousal state, an intriguing neural locus to consider was a population of neurons, visual projection neurons, termed the LC10A population, that were really initially described by a beautiful study from Ines Ribeiro, Axel Bors, and Barry Dixon as small spot motion detectors that convey retinotopically organized visual signals from the optic lobe, the lobular plate of the optic lobe, into the anterior optic tubercle within the brain. And these neurons, we know, are actually really essential for the faithful pursuit of males, as you can see here, because actually the kind of faithful pursuit of once these neurons are optogenetically inhibited, so silenced over time, males become basically, with the onset of the LED, become unable and basically blind to the presence of the female and are unable to continue to trace her with such high fidelity. Moreover, unilateral activation of these neurons expressing the light-activated ion channel CS crimson on one hemisphere or the other drives robust ipsilateral turning. And so what this indicates is that LC10A neurons are both necessary for courtship pursuit, and their activation is sufficient to drive the steering behavior that is required to track a female. Moreover, one really striking feature of these neurons is that we found that the visual responses of the LC10A neurons are strongly state-dependent. So there's almost negligible activity to the visual stimulus prior to the onset of courtship. But once males engage in spontaneous courtship pursuit, you can see that there's a strong enhancement of the gain of LC10A neurons, such that actually each time the visual target sweeps across the male's visual field, there's robust activation of the LC10A axon terminals within the brain. And notably, when males cease tracking the stimulus, even just very briefly before reinitiating, you can see that the gain of the LC10A pathway goes down back to its baseline, really highlighting that this visual projection population is modulated really on a moment-by-moment time scale. So if you look across animals, LC10A responses to the visual target are strongly amplified when males are courting, but actually not simply when they're running. And I think this is important because it indicates that the circuit changes accompanying courtship are really distinct from the more generalized forms of arousal associated with locomotion. This kind of specificity is further highlighted of this modulation by the fact that if we record from actually several morphologically related populations of visual projection neurons that actually sample from the same layers of the lobula and extend interdigitating axons into the anterior optic tubercle, so have axon terminals that are overlapping, what you can see is that these other populations of visual projection neurons are basically unchanged in the context of courtship. And what this really begins to highlight is the gain of the LC10A neurons is really kind of uniquely enhanced during courtship, providing a very selective conduit to drive courtship pursuit. And indeed, in accord with this idea, LC10A neurons we find form part of a very concise sensory motor circuit. And this is apparent from actually both transsynaptic tracing experiments that I'm not showing you in the male, but also analyses of the female hemibrain connectum, where we have synaptic resolution understanding of neural connectivity patterns now within the female brain of the fly. And what this analysis indicates is actually that LC10A neurons are connected to descending neurons through only 2 synapses. So there's a population of descending neurons that are actually only 2 synapses away from this visual population. Again, including actually descending neurons that have been implicated in steering behavior, like the DNAO2, which Rachel Wilson's lab was described as being an important contributor to the steering behavior of females. And indeed, actually, Adrian Otopalk has begun recording from some of these different descending neurons, really as an inroad to start to gain insight into the motor control of the different component behaviors of courtship, including tracking behavior. And what she found by imaging a number of these different descending neurons that are downstream from the LC10A visual projection population is that they are actually recruited during visual pursuit, suggesting really that the state-dependent modulation of LC10A responses, these visual projection neurons, are indeed able to propagate to the descending neurons to drive steering behavior when males are aroused. And so in further support of this, Tom Heinmar-Sten built a really simple network model based on the neural architecture of this circuit to see whether it could account for the visual pursuit of courting males. And to do this, he used the published spatial temporal receptive fields of LC10A neurons to structure excitatory input into this population of model LC10A neurons that really were able to tile across the visual space and then pooled into single downstream target neurons connected through actually a difference function such that the strength of turning to either the left or the right would be proportional to the difference in spiking between these neurons. And we find that actually this very simple network model is actually able to replicate a large number of kind of predict the behavior of males to a variety of different visual stimuli. So here you can see what you would have seen earlier if I had not omitted that slide is actually what the male fly sees projected onto the visual panorama, a dot that goes back and forth in a kind of an invariant manner. And what you can see as in the overlay of between the model activity and the behavior of the male is that when males are highly aroused, where we continuously activate the P1 neurons, actually the model replicates the behavior of the male to this very simple visual stimulus almost perfectly. So this provides us with some insight into the kind of the ability of this model to replicate very simple behavior, but actually also identified some more and kind of highlighted some more complex behavioral quirks. So for example, if we provide the male flies or the model with a visual dot that actually stops at the center of the male's field of view, the model actually is able to predict the behavioral overshoot you see every time that the visual dot stops. Or if we provide actually either the male or the model with two dots that move out of phase with each other, thus trying to mimic the more kind of complex visual environment that a male may encounter, like for example, on a rotting mango where there are multiple flies, you can see that actually the model is able to predict the accurate, the kind of strength and direction of the turning on a turn-by-turn basis as these two dots go out of phase with each other. So together, the kind of the predictive power of the model really underscores that we think that this pathway, the LC10A sensory motor circuit, is able to deterministically define the behavior of males when they're highly aroused. And indeed, actually, the model is also able to partially replicate the dynamics of freely moving males engaged in natural courtship, where female trajectories are obviously inherently much more circuitous and variable. And so to do this, we simply provided the model with the estimated angular position of the female onto the male's retina. And what you can see is that the model can predict the vast majority of the turns, even in free courtship. So taken together, what this suggests is that LC10A visual signals, once gated by male sexual arousal, are able to actually be able to propagate it to the descending neurons that drive steering behavior during courtship and really predictably drive turning in response to a moving visual target in an almost reflexive manner. But of course, I want to remind you that I told you that a male's courtship is not constant, but even in response to an invariant visual stimulus that is ceaseless, actually male's the courtship intensity fluctuates and wanes over time. And so we think that one of the key features to account for this kind of fluctuation in the intensity of male courtship is the fact that this visual motor circuit is under constant regulation by the intensity of a male's courtship drive that is conveyed by the P1 neurons. And this can really transform alterations in the moment-to-moment strength of a male's courtship drive to acute changes in the intensity of his pursuit on a moment-by-moment time scale. And we think that actually these signals about a male's courtship drive are actually emanating from the P1 neurons themselves, in part due to the fact that when we image the P1 population as males initiate spontaneous courtship, we find that actually their activity is tightly aligned with the onset of courtship in accord with the idea that these neurons trigger males into this long-lasting arousal state. But actually, quite surprisingly, what we found is that the activity of the P1 neurons is not representing this long-lasting state, but actually continues to fluctuate and reflect the ongoing fluctuations in the vigor of a male's courtship pursuit. And you can see this actually because there's a really nice correspondence between the activity of the P1 neurons and, for example, the tracking index of a male fly that reflects both the vigor and fidelity of his pursuit. And if you look across many animals, what we can see is that the P1 neuron activity is nicely and well correlated with a male's tracking index over time, but not with a male's overall kinematics, like the speed of the animal or the visual cue itself. And so this suggests that P1 neurons not only trigger entry into the lasting arousal state, but actually seem to surprisingly subsequently encode the intensity of a male's courtship drive. And indeed, if we actually perform dual imaging experiments where we record the activity of both the P1 neurons and the LC10A axon terminals within the brain, you can see that there's a really nice correlation between these two signals. And in fact, actually, the strength of the P1 activity level is really linearly predictive of the magnitude of the LC10A responses to the visual target each time it sweeps across the male's visual field, suggesting that the P1 neurons, in fact, regulate the gain of LC10A circuits in a completely scalable manner to control the vigor and fidelity of a male's turning behavior on a moment-by-moment time scale. So to further sort of test this idea that P1 neurons are actually actively and dynamically modulating the gain of LC10A pathways, we allowed males to initiate spontaneous courtship pursuit while imaging the LC10A neurons, and then actually acutely activated the P1 neurons optogenetically. And what you can see is that this optogenetic activation, the activation of the P1 neurons, both serves to enhance and boost the gain of LC10A responses to the visual target, but also leads to males to become much more vigorous and faithful in their pursuit of the visual stimulus. And so taken together, what this suggests to us is that our results really suggest a kind of basic model for how courtship is controlled over time, where males are basically essentially blind to the presence of the female prior to courtship initiation, where actually the LC10A circuit lies dormant and silent in the brain until a discrete moment in time when the P1 neurons become activated. The gain of LC10A signaling is enhanced and allowed to be propagated to the descending neurons that underlie the steering behavior of the male, and to allow for really a faithful kind of propagation of these signals to drive steering behavior in an almost deterministic fashion, giving rise to really high fidelity, robust courtship pursuit of a conspecific female. And so this feature of the courtship circuit, the idea that there's a really high fidelity sensory motor circuit that's under control of the P1 population, really begins to help us to understand how courtship pursuit can be so robust and invariant when males are in the presence of an appropriate female mate. However, we also find that the P1 neuron mediated arousal is not really a switch, but actually functions, we think, more like a rheostat to control, to really continuously control the gain of visual signaling, the flow of visual signals in a graded manner, depending on a male's courtship drive. And we think that this ongoing feedback is actually really critical to the logic of mate choice, given the evidence that I provided that actually visual acuity of males is actually very poor. And that is obvious because actually they will initiate courtship pursuit even to just a moving dot. And what this means is that, of course, that there must be additional input for discrimination to assure that males don't track other males or females of the wrong species. And so we think that this ongoing feedback, at least one source of ongoing feedback, actually comes from pheromone pathways, because work from a number of labs has suggested that P1 neurons, as I described, are directly integrating input from multiple pheromone pathways, including receiving input from inhibitory pheromone signals that can transiently suppress P1 activity. And so we think that this might suggest is that in the context that when a male, for example, is in the context of pursuing its conspecific female, as you can see here, he will pursue this conspecific female as an attractive target, become transiently distracted by the presence of a heterospecific female, taste the pheromones that coat her body that inhibit the P1 neurons. And we think that this might rapidly and reversibly lower the gain of the LC10A neurons to allow him to prevent continued pursuit of an inappropriate mate. And in further support of the idea that of just how critical this ongoing pheromonal input is to temper a male's visual pursuit, we can actually generate males that are broadly mutant for pheromone detection by mutating a critical co-receptor. And you can see that these pheromone mutant males, these males that can't detect pheromones, are completely indiscriminate and will pursue both conspecific and heterospecific females with complete equal vigor. And what this really highlights is that basically all fly-sized objects actually represent inherently attractive visual targets to males. And appropriate mate choices are honed not only by excitatory pheromonal cues that activate the P1 neurons to promote courtship, but importantly, inhibitory pheromonal cues that suppress P1 neurons to reverse the gain of LC10A signaling. Okay, so we've gone to gain some insight into sort of the dynamic control of male courtship through the kind of state-dependent processing of signals through this LC10A sensory motor circuit. But I just want to highlight a few of the ongoing questions that remain unresolved in this work. First, actually, I should say that we really don't understand the mechanism of LC10A gain modulation. And in particular, P1 neurons do not directly innervate the anterior optic tubercle, suggesting that even though they convey rapid signals about courtship drive to this visual population, there must be an intervening population of neurons that are able to transmit this signal to the LC10A population. And I think the selected gain modulation of this pathway compared to other visual projection neurons that are even overlapping really highlights and provides an interesting inroad to consider the differential modulation of visual pathways by the courtship state. Another question is that actually we don't completely understand how males become sexually aroused by vision alone, in part due to the fact that actually P1 neurons themselves do not appear to be visually responsive. And it suggests that there must be, again, additional cues or additional visual pathways that feed into the P1 population that might be actually more selectively tuned to the natural statistics of female motion that are actually going to impinge on this population and drive their arousal state. And finally, it's unclear how this long-lasting arousal state is actually perpetuated, since it doesn't appear to be encoded by the P1 neurons themselves. And so one possibility is that it is encoded by the activity of downstream targets. For example, one population of neurons identified by David Anderson's lab called PCD neurons, which are thought to be actually persistently activated once males initiate courtship, or as part of a kind of recurrent circuit that could actually kind of reverberate and continue to excite the P1 neurons themselves, or through potentially ongoing behavioral feedback to the P1 population. So there's still quite a lot to be done in exploring this mechanism, but I think it provides us with some insight into really how actually in one species, actually courtship pursuit might be implemented. And I just want to remind you that we think that the logic is fundamentally conserved across these different species, because as I mentioned, males of all species must become motivated to pursue a female in order to be able to actually entice her to copulate. And this raises the interesting question of what is the role that P1 neurons play across these different species. So to address this, Rory Coleman and Nessa Morante in the lab translated the selective genetic drivers that label the P1 neurons in Drosophila melanogaster into the genomes of several close cousins of this species. And what you can see is that it labels anatomically very similar populations of neurons. But I think the real power of this approach is it allows us to gain genetic control of the P1 population to demonstrate that they play a conserved behavioral role, because optogenetic activation of the P1 neurons across these different species all drives kind of indiscriminate visual pursuit towards inappropriate targets, in this case, either rotating magnets or even ladybugs. And so what this highlights is actually that males of all these different species, once become aroused, are actually willing to chase almost any kind of moving target. And this then further underscores the importance of additional species-specific cues that must be used to hone a male's mating decisions. Because, for example, you can see on this food patch how many animals actually, there are many appropriate kind of moving targets around, and males within this sort of more complex social environment must be able to determine which of these kind of moving visual targets is actually an appropriate mate and which of them are inappropriate mates. And we think that pheromones actually likely play a very important role and serving to focus a male's pursuit towards an appropriate mate, as I described before. And in part, this is due to the fact that actually female pheromones have really rapidly and repeatedly diversified across this genus under strong selective pressure, since mating between any of these even closely related species, as I described, produces inviable or sterile offspring. So as a consequence of the rapid evolution of female pheromones, even females of sister species, for example, Drosophila simulans and Drosophila melanogaster, produce different chemical cues that coat their bodies. And these chemical cues, these different pheromones, serve to both to promote courtship towards appropriate conspecific females, but it also importantly serve to suppress futile pursuit of inappropriate mates. And this raises a very interesting question. How is it that males of these two different species of melanogaster and simulans males, how can they detect the same chemical cues, for example, the melanogaster female pheromone, but endow it with the opposite behavioral interpretation in the context of courtship? where in the nervous system have changes occurred, where has evolution actually acted within the nervous system to give rise to these really opposing behavioral responses that underlie mate discrimination. And so to address this question, former graduate student in the lab, Laura Sholzer, actually performed a direct comparison of the homologous pheromone processing pathways in Drosophila melanogaster and simulans males. And what she did was to basically examine the anatomy, function, and behavioral role of neurons at each level of the circuit, from the sensory periphery all the way up through to the P1 population itself. And what we found is that the architecture of these pheromone processing pathways was really largely preserved across species. But actually, strikingly, the pheromone responses of the P1 neurons differed. So when a melanogaster and a simulans male taste the melanogaster female pheromone, in one case, it activates the P1 neurons, and in the other case, we see basically no activation or even slight inhibition. And we think that this differential response is actually due to alterations in the weighting of excitatory and inhibitory inputs to the P1 population. So what we think is a consequence of really what is subtle evolutionary tinkering within a conserved circuit architecture, the melanogaster female pheromone is transformed from an excitatory cue that activates the P1 neurons to promote courtship behavior into an inhibitory signal that suppresses a male's visual pursuit and his arousal. And so this first cross-species comparison, I think, was interesting because one of the things that it highlighted to us is it really kind of challenged the long-held conception that the sensory periphery might be the most favored target of evolution, and revealed really how central circuit nodes, like at the level of the P1 neurons, can serve as a substrate for behavioral divergence between species. Okay, so this first study was informative, and it provides us sort of with an anecdotal understanding of how really two species have diversified. But I think we're interested generally in understanding whether there are patterns or principles of how behaviors evolve that can only emerge from a broader sampling of the phylogenetic tree. And I should say that this is in part inspired from studies of morphological evolution that have revealed how really kind of, for example, enormous diversity can arise from actually repeated evolutionary tinkering of certain genetic loci, for example, the yellow locus, as shown by Sean Carroll's group, where actually repeated evolutionary tinkering in the expression of this one biosynthetic enzyme can actually give rise across different dipterian species to these amazingly beautiful patterns of pigmented wings. And so it raises the interesting question whether, just as in the context of morphological evolution, whether there might be neural circuit motifs that likewise may serve as sites of repeated evolutionary tinkering to facilitate behavioral evolution. And we don't know the answer, I should say, but we can start to begin to gain some insight into this potentially by leveraging the fact that across this genus, the same behavioral traits have actually arisen multiple times independently, including the distinct pheromone preferences that males use for mate recognition, allowing us to explore actually whether behavioral evolution proceeds in a predictable manner, for example, by acting recurrently at the same central circuit node to control a male's sexual arousal, or whether it acts at distinct sites within the nervous system through completely divergent mechanisms. And I should say that if we consider these four species, while I showed you that P1 neurons play a conserved role in promoting courtship across these species, we find that the functional responses of the P1 neurons to a panel of different female pheromones are highly divergent. And what this indicates is that we think that males of these different species actually all rely on really species-specific functional tuning of the P1 neurons to control their sexual arousal. And so we're therefore interested in examining whether the distinct pheromone preferences of these different species arise from the same sort of subtle reweighting of inputs to the P1 population, as we described in Drosophila simulans, suggesting potentially a convergent neural logic, or whether possibly every species has actually evolved a completely distinct solution for mate recognition. Okay, so this raises the possibility then that there might be inherently flexible nodes within the nervous system that are actually potentially the favored targets for behavioral evolution, like the P1 neurons. And this is something that we think is actually quite an appealing idea, given that actually, as we already described, subtle reweighting of inputs to the P1 population would allow for the rapid emergence of species-specific mate preferences. So while we began to gain some insight into the evolution of sensory pathways underlying mate discrimination, what I'd like to just highlight in the last few minutes of my talk is that equally striking is the diversity of behaviors that males perform once they become aroused. And if you look across the Drosophila genus, male courtship displays vary really dramatically in both their composition and also in their patterning, even between closely related species. And so, for example, in comparison to the intermittent bouts of song produced by Drosophila melanogaster males, actually Drosophila yucuba males sing in really very lengthy bouts, while Suzuki males actually don't sing at all, but rather flaunt their patterned wings in a silent visual display. And so we're interested to ask, to what extent are these species-specific courtship rituals reflecting the differential use of the same component behaviors, for example, to generate novel behavioral sequences, or do they actually reflect the evolution of completely novel motor elements? And so we are actually just beginning to approach some of these questions, but I would say that the LC10A, our work on the LC10A pathway kind of hints that there might be the different component behaviors of courtship are mediated in a kind of modular fashion, each by a parallel sensory motor circuit comprised of distinct visual projection neurons, potentially tuned to different features of the female. And then in part, this is due to from the fact that actually in LC10A silencing experiments, like you see here, where we optogenically silence these neurons, you can see that males are still able to actually engage in actually visually guided singing and orientation behavior to the female, even though they're no longer able to track the female with such high fidelity and precision. And so it's just that there must be other visual pathways that are involved in these additional component behaviors of courtship. Moreover, we believe that actually each of these parallel circuits might be differentially modulated by the intensity of a male's arousal state conveyed by the P1 neurons. And in part, this is due to the fact that in tethered courtship, we find that males actually are willing in their pursuit of the visual target, as males are pursuing this kind of invariant visual stimulus, that their willingness to actually track this visual target far outlays their willingness to sing to it. So they only sing for the first few minutes of this spontaneous courtship bouts that last for many tens of minutes. And again, suggests the possibility that these different component behaviors, tracking and singing, have actually distinct sensitivity to a male's slowly waning arousal. So this kind of modular organization, I think, really is kind of quite interesting because it provides a framework to understand the natural progression of courtship behaviors throughout the courtship ritual as a male's arousal escalates over time. And this is something that actually Adrian in the lab is starting to explore by recording, for example, from the distinct descending neurons that control the different component behaviors of courtship to explore how they're differentially recruited. So for example, some of the descending neurons I described before that are downstream from the LC10A circuit, like the DNAO2, are engaged, we think, in the steering behavior and mediate the steering behavior of courtship, where other neurons, like the male-specific descending neuron PIP10, is known to be essential for actually producing the wing motion vibrations that underlie the production of courtship song. And so by actually comparing the activity of these neurons as males are engaged in the kind of spontaneous and evoked courtship pursuit, will allow us to begin to understand how their male's ongoing behavioral performance of the elaborate courtship ritual unfolds and which actions he chooses to perform, how they're guided on a moment-by-moment time scale by both kind of external sensory signals, which in this case are invariant, but also by a male's changing internal arousal state. So we hope to get some insight into how actually the kind of complex patterning of courtship occurs with more than one species, but this kind of modular organization actually also suggests a very powerful substrate for the evolution of novel courtship displays. And in part, this is due to the fact that just by simply altering the visual tuning, for example, of some of these circuits could actually change and underlie, for example, Drosophila suzukii's idiosyncratic orientation to the front of the female, while actually modulating and altering the sensitivity of these pathways to the P1 neuron input in a male's arousal state could alter the patterning of courtship. And for example, give rise to species like Drosophila yucuba that persistently sing whenever they become aroused. And together, this, although this is very early on in our understanding of kind of the evolution of this sort of motor control, it suggests how maybe novel behavioral displays may emerge from tinkering with actually pre-existing circuit modules, and a model that we can actively test by comparing these sensory motor circuits across the different species in courting males. Okay. So I just want to end by saying, actually highlighting that one of the things that's been really exciting over the past few years, that it's a really fascinating and interesting time for comparative biology. I would say particularly comparative neuroscience, where we can actually start to leverage kind of advances in recent technical advances in genome editing to begin to gain insight into and directly compare the same behavioral circuits across closely related species and glean insight into how nervous systems adapt over evolutionary timeframes to generate behavioral diversity. But I think beyond shedding light on the evolutionary tinkering processes, through this sort of comparative framework, I think we can also start to gain insight into the functional architecture and computations of neural circuits that cannot be attained from studying a single species. And ultimately, by actually studying neural circuits through the lens of evolution, we hope to be able to start to tease apart which features of behavioral circuits are conserved and constrained across a lineage, and which are nodes, potentially like the P1 neurons, that are particularly amenable to evolutionary variation and can fuel behavioral diversity. And so with that, I'd like to just close and thank the members of my wonderful lab from a pre-pandemic era picture who carried out this work. I should say that much of the work on the kind of state-dependent control of male courtship and visual pursuit was carried out by Tom Hindmarsh-Sten, a graduate student in the lab. He was helped by Rufe Lee and Adrian Otapalik, who has also been working on sort of the starting to think about descending control of the different component behaviors of courtship. I mentioned Rory Coleman is another postdoc in the lab, has been looking at the diversification of pheromone preferences across different species, and has been helped by a senior scientist, Nessa Morante. We've gotten a lot of wonderful advice and input, both at thinking about genetic reagents and also about technical behavioral paradigms from David Stern's lab at Genelia, as well as Gabby Naiman's lab at Rockefeller. These are acknowledged sources of funding and support. And just to say again, how pleased and happy I am to be here today, even in my virtual remote format, and how I'd be delighted to take questions now, or if anybody wants to contact me, by email after the fact, I'd also be happy to talk about any of this work further. So thank you.
Thank you very much, Vanessa. I know you can't hear, I can hear our audience from where I'm sitting. It's a big round of applause. So the first question that we have is, speaking of the fidelity of the male pursuit, what happens to the LC10A firing in male behavior when a second female with tasty pheromones is interposed between the first target and the male, or when another male appears?
Yeah, so that's a really interesting question. One thing which we noticed is, so for example, if you give a male 2 equivalent females, he often actually quite faithfully pursues one, and occasionally if another female crosses his field of view, he will switch targets. And actually, we don't think that they can actually distinguish between 2 females with tasty pheromones that are similarly from two conspecific females. But there is one interesting aspect of the actually LC10A circuit that we started to explore, which is I showed very briefly, is, for example, if you give the male fly 2 dots that are slightly out of phase of each other, the male will actually actively or preferentially track one of the targets, which is moving in kind of a progressive motion fashion within his field of view. And so we think that through the sort of kind of lateral inhibition between the 2 hemispheres in the LC10A circuit, that there is actually some sort of preference for one of the targets over the other, just based on the visual properties of this pathway.
Is the activity or tuning of P1 different between virgin and mated males, or do you know their status?
Yeah, so that's also a very interesting question. So we haven't really explored this feature. I will say that actually Mike Crickmore's lab, amongst others, has explored the idea that actually one interesting thing about the activity of the P1 neurons is it's not only seems to be one locus for module or kind of differential regulation at evolutionary time scales, but actually even over the lifetime of an individual fly. So if male flies, for example, have never seen any other fly, they are extremely motivated to pursue actually moving targets. And in fact, that is one way that we can get really robust spontaneous courtship, is actually by keeping males isolated. But if males have had many sexual partners, if they've had many opportunities to mate, actually that courtship drive is decreased. And Mike Crickmore's lab suggested it was through neuromodulatory mechanisms that occur at the P1 locus itself. And so it suggests actually that it is also a kind of a node of regulation, even for past social history, as well as kind of evolutionary history.
I think if they're available, we have some questions from the students.
Thank you very much for the talk, Dr. Ruta. One question in particular, have you looked to see if sexual experience enhances the connections within this circuit, specifically for the P1 to the postsynaptic neuron?
Yeah, so actually, as I mentioned briefly, that so I think there is some evidence that past sexual experience probably does modify features of the arousal state, the courtship drive. And some of it is thought to be actually feeding into the P1 population. So for example, how pheromone signals are able to, as I described, there's sort of an excitatory and an inhibitory branch of inputs onto the P1 population and the kind of differential weighting of those two. We have not really looked very much at the downstream targets of the P1 neurons. It's actually one of the things we don't completely understand. As I mentioned, there are potentially downstream neurons like PCD neurons that David Anderson's lab has described that are involved in sort of maintaining the persistence and of a male's arousal state, but we actually haven't really fully traced out those circuits.
Hi, Dr. Ruda. Thank you for the talk. So I had two sort of related questions. Is there an upper limit for, or have you seen an upper limit for how long they will actually pursue a fictive female? Like if most of your scales ended around 15 minutes, but I'm curious if they'll go past that. And then sort of related, is there a, I guess you could call it a refractory period between courtship attempts where after which they'll return to that really vigorous P1 response at the start, or does that happen every single time?
Yeah, so one thing that we've actually observed is that in general, there is, even with either spontaneous entry into courtship where P1 neurons become activated, or even with transient activation of the P1 population, that actually it leads males to track for, I would say, tens of minutes. And we've gone up in our experimental work, you know, past 30 minutes. And there are males that will continue to track for that entire period. I think it's an interesting thing that we have not explored is whether you continuously reactivate the P1 neurons, as it probably happens in the context of natural courtship. And maybe that's the role that female, like conspecific female pheromones play, is to actually continuously reinvigorate the P1 neurons and actually extend out that time scale. But we have not explored. I think it's a really interesting question, what actually maintains and sets the time scale of a male's arousal, and actually, you know, how does it ultimately decay in the absence of any other kind of sensory or behavioral feedback? I'm sorry, there was a second part of the question which I may not have answered. But...
Curious, do they always return to that vigorous initial P1 response right after, or is there like a refractory period?
Yeah.
Or something?
So I think, again, we haven't completely explored it. I think with artificial activation of P1 neurons, I think they do, and they do return to a highly elevated state of courtship. And actually, you know, you could sort of see that in experiments where we were imaging the LC10A neurons, that even when males initiated courtship spontaneously, you could see that their arousal decayed over time. But then if you activated the P1 population, you could boost males right back to being really highly aroused. And so it is, you know, in some sense, males are kind of, I think, really primed to court, and they're really only waiting for inhibitory cues that suppress that kind of intrinsic inherent drive to identify or find to mate and to pursue a mate.
Hi, thanks. I had a question about the pheromone page. It seemed like a lot of the species did have the same pheromone, at least it was labeled that looks similar.
That's right.
Do those species live in different areas? It also seemed like some of the species had stronger response to their females' pheromone cues than others, and it seemed like maybe some of the species with similar pheromones didn't have as strong of a pheromone response. Did they have a visual or behavioral difference that they are more responsive to as a consequence of their pheromones being fairly similar?
Yeah, so you asked many good questions about the logic of pheromone, you know, mate choice based on pheromonal cues. So I think one of the interesting observations is actually there are a number of species where the same pheromones are used by females. And in general, we think that most of those species actually, one of the major drivers of pheromone evolution, as described by other labs as well, for example, like Sean Carroll's lab, has been actually that those species are overlapping in their environments. And so there's a strong selective pressure to produce, you know, different chemical cues that would signify species identity. So for species where females have the same pheromones, we think very often that they are not actually they often don't encounter each other in the environment. And at least in the laboratory context, for example, males very often cannot discriminate between females that carry the same pheromonal cues and which would indicate actually how important those signals are. Like there's no other signatures of behavior or even visual differences that differentiate between them in preference assays. And so, yeah, so we think that chemical cues are really very critical. You asked also about the activity level of the P1 neurons, and there are quite a few nuances there that are kind of interesting to consider. One thing which I didn't get into at all, but actually is an interesting feature, is that also, I guess, highlights the importance of inhibitory cues and kind of interplay between visual arousal and inhibitory cues, is that there are a number of species where actually males and females carry the same pheromonal cues on their body. So they're monomorphic in their pheromone production. And we think that therefore they're kind of basically, they're non-informative for mate discrimination. And so in that case, they must use additional signals that can distinguish between, distinguish between the sexes. But I guess it really highlights the idea that those cues we think very likely are not themselves that informative to promote arousal. And actually in that case, probably males rely mostly on visual arousal and then inhibitory pheromones to suppress that arousal towards inappropriate mates. So it's kind of an inverted logic to what we often had thought about, I would say, in terms of mate discrimination from maybe other species.
Got a couple more that have come in on the chat. Do female flies have P1 neurons or the equivalent cells that alter receptivity? And might these also serve as nodes for evolution that are activated by different displays?
Yeah, so that's a great question also. So as I mentioned, P1 neurons, which have been described for a number of years since their first identification by, for example, Daisuke Yamamoto's lab and others have suggested that these are really male-specific neurons. They are population that are actually just not present in females. But there are kind of an equivalent population called PC1 neurons that are thought to be involved in controlling a female's sexual receptivity. And I think these are actually very interesting to think about in terms of what regulates their activity. Recent work from Barry Dixon's lab has highlighted that, for example, like the male courtship song is supposed to be the most important signature of a kind of sensory cue that females use to identify an appropriate mate. And so those neurons, for example, are in part tuned to the kind of statistics of the conspecific courtship song, the courtship song of conspecific males. I think one interesting question is sort of like what really females pay attention to. And I would say that one of the challenges, I think, of female behavior is that actually females seem very passive in their role, especially in melanogaster, Drosophila melanogaster, where actually most of the time we measure receptivity as just whether or not the female is willing to allow him to copulate. And that doesn't, that's sort of like an endpoint assay where it's very likely that there's also a very dynamic process we imagine that could be involved in, internally, a dynamic internal arousal state that is sensitive to the cues of the male and the performance of the male courtship ritual, but we just don't really have a good sense of how that maps to controlling her arousal on a moment by moment time scale. So I think that's a very interesting direction for the future to kind of explore.
And do you know what neurotransmitters the P1 neurons synthesize?
So that's also a very good question. We've actually done some sequencing of the P1 neurons just to try to understand better. For example, like, is there an obvious kind of molecular handle that could provide us some insight into like what would underlie that kind of lasting arousal state? We know actually, I think they're in part cholinergic, which is the excitatory neurotransmitter that's often used in the central nervous system of the fly. But they also express a lot of neuromodulators and neuropeptides, which I think are interesting candidates that could, act on downstream circuits to kind of amplify and perpetuate the kind of arousal state through, by kind of modulatory changes in the downstream targets of the P1 population. But I would say that we don't have a very good mechanistic understanding of that process just yet.
All right, we have one last question. Does the cross-species variation of the relative importance of pheromonal, acoustic, and visual information correlate to the different environments which courting occurs? It's hard to get attention with one's voice in a noisy bar, but perfume would work.
So that's a great question. Because I think there are going to be very interesting differences across species in the relative importance of different cues that depend on also their ecology of the different species. And I can just give you one example from work that I didn't have a chance to talk about from a postdoc in the lab, Philip Brand, who's been thinking about how one species we've looked at, Drosophila erecta, for example, will only court on the presence of a food substrate. So they are a specialist species, and their arousal seems to be strongly gated not only by female pheromones and by visual cues, but actually by olfactory cues that arise from the kind of the food that they're actually often meeting their mates on. And so it highlights how actually the different sensory inputs probably think to the P1 neurons or to some other node within the nervous system are really kind of fine-tuned across different species to assure that males become aroused only in a kind of appropriate, ecological and social contexts. And that's something I think that will be fun to explore going forward.
Thank you, Vanessa, for being a wonderful speaker tonight. Give a in-person and virtual round of applause.
Thank you so much. I really appreciate the chance to be here. And
this concludes the lecture portion, but everyone is welcome to stick around for the live town hall.
Hello, everybody. Could people see my screen? Could you see my slides? Could people hear me? I think I'm okay. Wait. So good morning and welcome to NSNB 2020. My name is Dr. Gravid Leachybrow, and it's so great to meet you guys. But first, let's start out by just introducing ourselves and get to know each other just a little bit.
Sorry, I'm just working on some stuff. I'll be here in attendance. I'll make sure to pay attention to as much as I can. Yeah, I'll follow along. No worries.
Hi, I'm Cynthia. I'm a mom working from home, and it's been very difficult lately. Michael, can you go find Daddy? I'm really busy right now. Sorry, guys.
Hi, hey, hello. Yes, I'm here. I'm super excited to be here. Looking forward to working and loading up on sleep whilst I'm here. I heard we work really hard.
But I heard we play hard too. Whoa, NSNB.
Hi, I'm here, but I'm just really bummed that I can't come and try popovers. People keep on telling me how great they are, and
Nala, I'm trying to introduce myself. Like, could you move, please? I'm sorry, everybody. This is my cat, Nala. She thinks she's part of the meeting.
Hey, kitty.
What is happening? Do people hear me?
Can you be quiet?
Is everybody there? I think we got so much to do today. I just want us to get this, completed so we're not here until 3 a.m. So, let's go over some ground rules. I hope everybody read, all the things we sent. I think we sent them yesterday. They should be, about 20 hours of MATLAB, maybe 6 books, a few documentaries. I hope everyone had a chance to go over those real quick. But, just number one, there are secret beaches at MBL. Don't go to these beaches. Next, the Woods Hole experience, you want that to be accurate. So we just want you to just modify some things at home just so that you get the real feel. And one of the things that, we find that works really well is that you make sure your pillows are nice and lumpy, and, make sure your blankets are really thin. So, open your windows too, so you're nice and chilly. Okay, we're gonna learn about the leech ganglion, and we're gonna record from some neurons. So if you guys could use your annotate tool, where's the annotate tool? I think it's up on the, I don't know where it is. I think you guys are young, you guys can figure it out. And let's try to find a P neuron. Everybody try to find a P neuron.
Oh no, I think I'm on the wrong side of my leech prep.
Good attempts. Let's balance the bridge. You must balance the bridge. Look at that.
Oh,
perfect bridge balance.
Oh, I can almost see pie in the sky.
I don't know what balancing the bridge does, but I've been told it
really doesn't matter?
I think we used it to, Michael, can you just?
Oh, let's graffiti the bridge, huh?
I think we're like running low on time. So, I mean, let's go on to the next thing. So try and find the difference between these two rigs.
Oh, the bridge isn't balanced on the right one.
But anyway, they're exactly the same rig. So thanks for playing.
One eternity later.
So congratulations, everyone, on making it to the end of the first module. Wasn't it great? So it's time for the end of cycle party. Time to show off your moves. Don't drink too much, but you could drink plenty. So feedback. So, we want to try and make this program, better we just would like your feedback.
Yeah, I mean, I was didn't appreciate having leeches sent to my house. Maybe don't do that next time.
I wanted to party with you all.
I think the course is pretty great, but I would be much more engaged. Sorry, guys.
I will really miss my cats, but I think it would be easier to learn without them trying to be a part of it.
I'll definitely be sure to bring my own pillow and blankets, and then I'll be set to party.
What about the popovers?
Maybe we should just cancel this year. We could try this again. We'll keep you informed. We'll send you emails, and
1 year later.
Hi, everybody. Welcome to the NSMB Town Hall. And we are here with all the students, and I thought it would be nice to share with all the NSMB community that we are back broadcasting from the laboratory. So I'm gonna pass the microphone to the students to introduce themselves briefly.
Hi, my name is Catherine Newbury, and I am a third-year graduate student at the University of Pennsylvania.
Hi, my name is Rachel Hoyer. I'm a postdoc at the University of Miami.
Hi, my name is Sandra Cardenas. I'm a research trainee in Albert Einstein College of Medicine.
Hi, I'm Priscilla Yebu, and I'm a third-year PhD student at Stony Brook University.
Hi, I'm Cesar Medina. I'm a sixth-year PhD student at the University of Arizona.
Hi, I'm Kira Shapiro. I'm a sixth-year graduate student at the University of Pennsylvania.
Hi, I'm Chad Moon, and I'm a third-year student at Rockefeller University.
Hi, I'm Sun Myung-sun. I'm a third-year student at Harvard University.
Hi, I'm Maddy. I'm a postdoc at the Salk Institute.
Hi, my name is Annie Park, and I'm a second-year postdoc at Oxford University.
Hi, I'm Emily, and I'm a third-year grad student at Caltech.
Hi, I'm Dylan, and I'm a third-year PhD student at Dalhousie University.
Great, and we also want to acknowledge the intrepid and amazing faculty and TAs who came to take part in this live 2021 NSMB. And now we'd like to invite some people, pre-invited guests, to join us and say a few words about their NSMB experience and their relationship to it.
Yes, perhaps we should start with Eduardo Macaño, one of the first directors of the course, and wearing a NSMB t-shirt from the 19th century.
Well, it feels like it was that long ago. It certainly was another century. In fact, I was the director of the course. Well, probably well before many of the students were born. 1981 to 1984. I must say that some things look the same, but some things are, of course, totally different. a talk like Vanessa's was only imaginary, that we could possibly do something like that with a single neuron in a fly. I mean, it was hard enough to do something with a single neuron in the leech. And as you know, of course, a fly can fly inside the leech without hitting any walls. So anyway, it was a grand time because the beginning of this new phase of rapid changes in neuroscience was clearly happening. And it has resulted in tremendous advancements that the students these days can benefit from. Anyway, it's a pleasure to imagine being there, and sometime I hope I'll be able to come and see a class happening.
Next year, you're invited to come next year.
All right.
Something with you, Eduardo. How about Catherine?
I saw Eduardo and I ran off and got my 2004 NSNB t-shirt. I think here we go.
Yeah.
I have a good selection. I'll bring. So I was a course director a little bit after Eduardo Macaño, along with Rick Levine from the University of Arizona. I think we were directors from 2000 to 2004, I think. And some of your faculty took the course when we taught it. The NSNB, you've heard, it's famous for turning out fearless experimentalists, and we're so happy to see you. And Vanessa, I loved your talk. And thinking about John Nicholls, who was always a friend to the course, he would have been very happy to hear it. I'm going to be coming to Woods Hole for a Grass Foundation meeting, but I fear it's after your course is over. But I hope that I will get to see you in person at perhaps one of the meetings of the Society for Neuroscience.
Yes, and again, next year, we're expecting you to visit.
Oh, yeah.
So,
by the way, I learned about the leech by taking the leech course in Cold Spring Harbor from John and Ken Muller and a group of others. But John was a fantastic teacher, always around. But since I was a physicist, I came, to go into neuroscience from physics. I took one of the courses I took was that course on the leech, and the first thing I did was to look at the rig, and the oscilloscope was out of kilter, so I took it apart. And immediately John was on my back saying, "You can't destroy the rigs, we need the rigs." And I said, "John, I've taken apart and put together oscilloscopes many times." And so he allowed me to continue, and the course was fun. Anyway, John Nicholls, great guy.
So Andre, are you there?
I'm here. Can you hear me?
Yes, there you are.
And I'm wearing my course T-shirt from 2015, which it doesn't look very remarkable this way, but it does that way. So, welcome everyone. It is wonderful to see you there. I'm afraid that I will arrive on only Sunday and be able to wander through the laboratory. And I've in fact actually already done such a walkthrough about 10 days ago, because I was setting up a Whitman laboratory. So you will see me around. I was a course director from 2013 to 2018. And I have to say, before I became a course director, I knew nothing about NSNB other than it was a poster that was posted on the occasional bulletin board when I was doing my my PhD. And I had very little interest in it and very little knowledge about it. And in being recruited to become a course director, I absolutely said, there's no way I'm interested in doing anything like this. I couldn't imagine why I would be interested. And the wisdom of Jim Knierim in particular, who invited me to come, he said, well, there's something about NSNB. It's kind of magical, Andre. You really should come. Why don't you just come for 2 days? And reluctantly, I agreed. And I have to say, within the first half day, I called my wife and I said, there's something really like special that happens here. And I have to say, within 2 days, I could see something that absolutely made me committed to NSNB. And that was, I saw live in real time how the trainees, how the students, the people in the lab right now, how they transformed. And so the theme of my directorship became really embracing the idea that NSNB, helps to transform trainees into scientists. And you could really see it. It was during the Leach module, and people started out sort of fumphing and wondering, what they were actually doing with their electrodes and not really understanding what a voltage clamp was. And within 2 days, they were speaking eloquently and intelligently and insightfully about the biophysics of membranes. And it was truly remarkable. I have trained a lot of people in my laboratory to have that kind of facility, and it takes months, years. Some never catch it, and you could observe it happening in real time in 2 days. And so I became totally committed to NSNB purely out of selfish reasons, because I wanted to know, how do you do this? What's this magic? What happens? And then I realized what it takes. It takes, working 20 hours a day. It takes absolutely dedicated faculty, and the whole environment, the whole infrastructure of being set up so that you can do whatever it is that you choose to do in an absolutely fearless fashion. Right. That's the key thing I think that's wonderful about NSNB. It shows, and it in fact has made in some sort of standard and reproducible way, this magical thing that happens every summer where 20 students, this year it's a little bit less because of COVID, but where 20 students get to engage in fearless science. They get to engage in doing something that they've never done, that they're going to try to discover in the span of 2 weeks. For the first 5 days of those 2 weeks, they're going to learn the thing, and then they're actually going to be let loose on it. And that sort of magical and transformative experience is what makes great scientists. And so I am totally jealous that I didn't have that experience. I am absolutely jealous of it. But I am also extremely grateful to have been part of the NSNB family. And I toast all of you. I hope you capture and recognize, you probably won't because you're not sleeping enough. You probably won't recognize it for now, but you will for the rest of your careers recognize how important this experience is. And please continue doing this kind of science and this kind of training within the community. Congratulations and have a wonderful time.
Thank you. Thank you so much, Andre. And now we would like, can we hear? Oh, Catherine.
I forgot just one more thing. I know everybody is working as hard as they can and sleep deprived, but you haven't lost your sense of humor. I love that skit. It was hysterical.
Thanks again. And now we'd like to hear from two former NSNB students also on the call. First, 2019 student Cara Fulton.
Hi, everyone. I'm super excited to be here. As Steph mentioned, I was a student in 2019, and it definitely was a transformative experience for me. even now when I think about some of the limits that I thought I had during NSNB, they were challenged almost daily. You know, you go in and you don't think you're going to be able to accomplish a couple data slides after a full day's work of experiments, and then you are able to just kind of push through and present your data the next day. And I found that throughout the course of NSNB, I wondered if maybe this is the limit, and you are going to find for the rest of your experience at NSNB that you're just going to keep pushing that limit. And now that it's been, I guess now 2 years since I've been at NSNB, in my current experiments, I'm still constantly reminded that, this is like a tiny struggle, but we accomplished so much during NSNB, and it's reminded me of like how creative you can be, and exactly what multiple people have said already, that you can be fearless and ask questions that are maybe a little bit crazy, and maybe it won't work, but at least you kind of have the satisfaction that you tried. And that is something that I will take with me throughout the rest of my career. And it definitely has changed the direction of my career. Actually, I'm now joining a behavior lab for my postdoc, and I didn't expect that. So it's something that has definitely changed the way I think about science and the way I do experiments, the way I ask questions in lab meetings or in seminars. Just having this like fearless attitude that just try it, see what happens. I think that was something that I didn't realize was missing until I found it. So I'm excited for the rest of your summer. I guess you're almost halfway, and it's that that turning point between cycles. So I'm excited for you of what's to come. It was definitely a life-changing experience for me.
Thanks so much, Kara. Yeah, we're in the home stretch here because we had to shorten it from 8 weeks to 5 this year. So they've only got a week and a half left, but anyway, so next year, hopefully the whole 8 weeks. And last but not least, we'd like to invite 2019 student Eric Edsinger to come on.
Well, hello, I can see Molly, and want to say hi to Molly, and Molly can see her bench space, which I've positioned myself so she can check on her worms. And then Kara, so good to see Kara, who I just saw a second ago. Wow. So I don't know, I don't have any words or thoughts ahead of time, but one of the things that I'm recalling right now is I am kind of now, but I am on a neuroscience or behavior paper with Octopus with Gould Dolan. And in the process of that, I was marine biology, developmental biology background. And I went and visited Gould, and at one point, she just made a comment of something about, you really need training in neuroscience. I could kind of she could do the experiments and I could kind of understand it, but I didn't have any formal training. And just kind of off of that little comment, I thought, oh, I wonder where I can go do that. And of course, I was at the time a postdoc at the MBL, so it wasn't very hard to kind of connect the dots and think, oh, I should take the NSNB course. But even though I was at the MBL and I'd already seen the courses go through, I think at least 1 year already, it didn't really register for me. And I applied to the course, I got in, and I thought that was already pretty amazing because I don't have a neuroscience background. I was just kind of exploring it and wondering. And so they kind of let me in that sense. And when I started the course, it transformed my view of science, the MBL, where I had already been for 2 years, and just so much of I don't know, I can literally be 3 in the morning with Kara and a rig, and it just, everything's falling apart, but we're gonna try one more time. And that kind of crazy exploratory, just go for it, was really, yeah, it makes a big impression on you. But faculty that are just so accessible with their ideas and their models and their excitement, and I really felt like at the end, I just kind of walked into neuroscience, you know, into neurobiology, and it was a very welcoming and really beautiful experience. So that was really fantastic. And then along the way, I personally fell in love with Hydra. And so we have on my bench right now from the course, Hydra that I worked with in the course. And I'm continuing along some of the ideas I got from the course, actually from my little project, and going forward with that. So, you know, it has an emotional impact and kind of guides your thinking. Definitely brought me into the field. And now I'm sitting, I think like Kara said, she's in a new direction from taking the course. And I'm literally sitting with animals from the course. So it was a really profound thing.
Great. Thanks so much, Eric. I'm going to open to the students if they have questions or comments to the people that join us. No? Yes, Stephanie.
So when Eric says he's sitting with animals literally from the course, you may or may not know that Hydra are immortal, and he may have Hydra from the course.
Literally from the course.
Okay, so I think that one of the things that we want to do is first thank the NBL, Richard, Eric, and Ashley for helping us to put this. This is amazing what they did. They're very involved with this crazy idea, but I think it worked very well. We are the first course trying this. It's copyright, so hopefully it's going to have some revenues for the NSMB. And we would like to end with a toast for the future of NSMB. And of course, without alcohol or beverage, I mean, to observe for the guidelines of the COVID. Okay, for NSMB, I hope to see you all next year joining us. Okay.
Woohoo! I drink to you, for you. Cheers, folks.
Thank you very much.
Are you headed to the kid now?
You know, the kid closes at 9 o'clock. Do you believe that?
There's a pandemic happening.
No, it's insane. It's just
What a shame.
unbelievable. Anyway,
Well, that's what we usually used to do at midnight anyway.
Anyway, thank you everybody for joining, and I hope to see you all next year. Bye-bye.
Bye, guys. See you in a few days.
Okay, bye, Andre.
Thank you, everyone, for attending.